For the kind introduction, and I'm happy to share our recent publication with you all. So today, let me dive much deeper into the spatial thymus cartography that we made with the fetal and pediatric thymus. So as you can see, you can map the tissue quite well spatially. So what this allowed us to do is also geoposition lineage transcription factor in rare mimetic thymic epithelial cells, which I will be speaking in detail later on. So for the agenda today, I will start with explaining what is thymus and what is thymopoiesis, and move on to how we perform spatial profiling using StereoSeq, and then a definition of thymic niches and how we geoposition the cell types within it. And thymus is a site of T cell maturation. So there is an ongoing positive and negative selection of thymus that I will be explaining in a much more detail. And then finally identifying lineage transcription factors in rare mimetic TECs. So thymus is an organ that is present near lungs and actually behind the heart. It is the primary site for T cell maturation. And as I mentioned, the T cells are matured and selected within thymus, and it is crucial for the development of adaptive immunity and central tolerance. So as you can see here, the thymus is a structured organ where you have the outer capsule encompassing the organ itself, and then you have cortex, outer layer, and the inner medulla. And these are connected with blood vessels that make up the septum. So here is a H&E staining where can already with the staining itself see the different regions from the outer cortex to the inner medulla region. So next, moving on to what is thymopoiesis or T cell maturation in thymus. From the bone marrow, the progenitor cells, known as thymocytes, migrate into the thymus through the cortex and then go through the medulla, and then finally you get mature single positive T cells. So in cortex, the positive selection of the T cells takes place, where the negative thymocytes, double negative thymocytes move on to become double positive thymocytes with both CD4 and CD8 positive. And then due chemokine selection basis and with the help of the cortical thymic epithelial cells, these become more mature and finally enter the medulla region as a single positive thymocyte. Here, further, what happens is there is a diverse pool of antigen-presenting cells, including the medullary thymic epithelial cells, dendritic cells, B cells, and also a special class of cells called the mimetic cells, which represent different cell types within the person or different organs to train the thymocytes and for autoreactivity. And here, these cells are present antigen, and the main purpose is to eliminate autoreactive thymocyte and finally get the single positive T cells that are either CD4 positive or CD8 positive. So next, to understand the development process in thymus, we perform spatial profiling using StereoSeq T-chip. This is a spatial unbiased whole genome transcriptome profile. And what the StereoSeq is DNA nanoball array-based technology where it's an in situ RNA capture. And the capture is done in these spot size, right? So each one is a bin, and then there is just a bigger square for different bins. So basically, you can more or less select the cell type size you want roughly For instance, fibroblast, which is around 28 micron in size, a bin 50 would be much more closer to that. However, for an epithelial cells in thymus, such as the cortical epithelial cells or the medullary epithelial cells, the size roughly ranges from 9 to 17 micron. And for these, we might have to go down to a much higher resolution of bin 20, which captures the 10 micron size. And for the T cells, for instance, are quite small with 5 microns. These are, we need to go in higher resolution to even study the T cells. And this is possible by going down the number of bins that you select. However, one of the major challenges with an immune organ, especially such as thymus, is that it is very densely packed, making cell segmentation one of the very difficult tasks where you don't get the proper boundaries of the cells, which is much more easier to get from a brain tissue, right? So in thymus, because each of these epithelial cells are surrounded by many T cells that are quite densely packed, it is a bit difficult to get individual cell resolution with the cell boundary itself. So in spite of that, we wanted to see how during development, especially how the thymus is formed. And in this study, it is quite crucial to see the fetal and the pediatric thymus, because thymus is known to be got decrease in the thymus output from 2 years onwards. So the main development happens before that. So here we collected post-conception 13-week, 14-week, 17-week, and 18-week human thymus, and also from pediatric population with 7-week, 2 years, 5 years, and 6 years. Thymus is one of the organs when you do a heart surgery is usually taken out because after 2 years it is not much of an output in sense. So there is still survival. So you can it is something that is discarded. So that is how we collect the thymus samples from the hospitals. So next, what we did was we performed single cell RNA-seq to find cell types that are present, various different unique cell types that are present in thymus, and also the marker genes associated with that. And we also performed spatial profiling for all of these 8 samples. For these 8 samples, then we can spatially cluster them based on similarity in the transcriptomic profile, spatially cluster these spots. And further, we group the spatial clusters using the cell marker genes, and then we integrated across the fetal and the pediatric samples to form the high resolution spatial atlas. And then we could go further to look at which are the cortex, medulla, or septum. And here, what we show is the section view of thymus. We used a 1 by 1 centimeter slide, and for a fetal thymus, this is enough to capture one whole lobe. So for instance, here the 13, 14, and the fetal 18 week is one whole lobe of thymus that we can capture, which means all the tissue is intact, more or less. Whereas for the pediatric, the thymus is quite bigger, so we take a smaller piece and then profile them spatially. So here at bin 50, which is 25 micron size, this is the number of spots that we were able to capture for 13 to 18 week. It is the exact size of the one lobe of thymus. For the pediatric cases, it is a smaller piece. We also wanted to get as much as tissue intact. So for one of the samples, we did a 2 by 3 centimeter to capture around 300 K spots, whereas the 1 by 1 usually gives the maximum around 100 K spots. This particularly is important if you want to do find some rare events that occurs in the tissue. So then looking at the quality of the number of genes that is captured across the tissue here. So we see around nearly 1,000 number of genes present in like the 13-week fetal samples. And for some of the samples, we do see a bit of heterogeneity in the number of genes that can be captured, going down to 250 genes for the very large tissue. And also, it's much more difficult working with the pediatric samples for the quality reasons. So we went on to integrate a different quality samples together to make inference on the thymus and thymopoiesis. On the advantages, we can directly visualize the cell types based on marker genes on the tissue itself and the positions. For instance, T cells are present actually throughout the thymus. The cortical TECs by this marker is present mostly in the cortex, and the medullary TECs are given by AIRE is present in the inner regions. So as you can see, the complexity of thymus also changes, where only one lobe has one lobule, whereas in the 14 weeks, you get 2 or 3 lobules. And by 17, 18 weeks, you have many, many lobules within the thymus. It says lobules are the smaller structures of cortex and medulla, cortex and medulla. So next, we went on to define the thymic niches across the samples so that we can compare. For that, first, we built an integrated single-cell RNA-seq profile from the samples from this study and also public data. So this, the UMAP here shows all the different cell types that are present in thymus, where you can see most of them are double positive progenitors and quiescent state double positive thymocytes. And these, in the sense, mature to a single positive state. And other than the T cell itself, we have the thymic epithelial cells, MTC1, 2, 3, 4, and also some of the mimetic TECs like myo TECs and neuro TECs. And then there are other cell types are the smooth muscle cells, endothelial cells that makes the blood vasculature around the thymus. And here is the set of marker genes for all of the different cell types. So what we did for the spatially inferred clusters, we first did latent clustering and clustered the spatially similar transcriptome spots together. And then because there is some dropouts in different quality of the number of genes that is detected, we wanted to really make sure that the quality of the assembly doesn't reflect in the integration. Hence, we chose to use the cell type marker genes alone, which comprise all the cell types that can be detected within thymus, and then do a pseudo bulk for each of the cluster for these genes alone, and then did a PCA PCA and k-means clustering to recluster these spatial clusters across the sample. And finally, we can assign these to cortex, medulla, and septa based on like a CTEC or MTEC markers. So in the end, we have a much more comparative thymic niches defined here with green as septa, the blue is the cortex region, and the innermost is the medulla. So already here you can nicely see based on the colors that 13 week you start with just one lobule and then it slowly increases to 18 week. And then after 2 years, there are some tissue changes in the thymic architecture itself. And we further divide the cortex into 3 regions because thymus is the T cells maturations are in stages. It goes through the cortex and then through the medulla. So we divided the outermost as cortex 1 and the innermost cortex as cortex 3. And then similarly, medulla 1 to medulla 3 as the innermost one. Then we do have a single cell transcriptome profile. We know what are the cell types that are present in the thymus within these samples. And because we can directly look at marker genes from the spatial, we can do sort of a scoring for these marker genes and then find that the double positives and most of the thymocytes are present in the cortex 1 to 3 here. And if you look at the medulla region, the cell types that are present are mostly single positive, like the CD4 positive T and the CD8 positive T cells. And also, some of the medullary T cells are assigned to the medullary region. And then in the septa, we find other cell types like the endothelial, smooth muscle cells, and so on. We also found that there are low quality regions that the transcriptional output is very within the tissues itself. And for some reason, we did see a very high in MTEC or medullary TEC. So I will explain this feature a bit later in the slides. And so this is based on transcription profile. We wanted to see if we can recapitulate this with a few protein marker genes, and we did maxima protein profile for a selected few marker proteins, and then like CD8, CD4, and you can see where the T cells are, where the miniature killer cells, which are more present in the medulla, and dendritic cells present in medulla because they are the antigen presenting cells, and further in the septum, we have fibroblast. And this shows that they are comparable, and you can define thymic niches based on just transcriptomic profile alone. So in the next module, we have defined our niches, and we wanted to know how the positive and negative selection of T cells occur. So as I've shown you before here, the double positive the thymocytes, the mature thymocytes enter the thymus and become double negative thymocytes, become double positive. And then these are mediated by some chemokine pathways like the CCL and CXCL here. So when you look at the cortex, and then we did cell-cell communication analysis, we found the CXCL to be quite active in the cortex region across the sample. And then, as you can see here, the CXCL, the receptor, and ligands, along with cortical TEC epithelial cell marker PRS16 is present over the cortex region. Further, I did mention previously that the fetal thymus is a much more intact organ, and we sequence the whole organ intact, at least one lobe, the whole one lobe as it is. So the reason it also has the capsule that can be defined. For instance, the outermost region is a capsule, and you can see along there is a fibroblast layer along with the MDK, MK pathways. Next, we want to look at the signaling pathways that are active during the negative selection of T cells. Here, as expected, we do see the CCL pathway, and it is more concentrated within the medulla, which shows a very clear structure here. For reference, the cortex is in the outer region. There's the capsule region, you have a outer cortex region, and just looking at the expression of these genes itself, you can define these are the medulla region. And other antigen presenting cells are also present and are involved we use the marker genes of MS4A1 along with the MIF pathway. It also shows a clear definition of the medulla niche. In addition to this, there is also galectin pathways. The galectin is associated with cell death during the negative selection of the T cell. And some of the T cells that are autoreactive gets terminated, and the macrophages are acting on it. So these pathways are quite important in clearing the T cells that are not selected. So next, we could recapitulate the thymic niches. We can explain some of the thymopoiesis signaling pathways that takes place, and I'm wondering if there are other rare mimetic TECs that are present in the spatial context that we can identify. Because even in single cell, when you do it, you have to really enrich for EpCAM time epithelial cells to get them. So as this is an unbiased approach, we try to find if there are any rare mimetic disease. So a quick introduction on the mimetic TECs. The medullary epithelial cells within the medulla expresses some other lineage transcription factors that are not related to thymus at all. So the main purpose of doing this is only to train the T cells that do not attack its own cells that occur in other organs, right? For instance, these mimetic TECs mimic peripheral counterparts such as muscle, shown here, through an expression of the MyoG transcription factors. So this mimetic TEC that is within the thymus expresses MyoG, and when it expresses the MyoG transcription factor, the peripheral antigens are also expressed. So when the T cells come and then try, if it recognizes this myogenes, mimetic TECs, they will be terminated because it is autoreactive. So this is a selection process that all the T cells has to go through. Now, the role of the mimetic T cells is to present the T cells with as many possible cell types that you can find within the person so that it is not auto reactive and it doesn't trigger autoimmunity. For instance, these are some of the known ones where, so AIR is one of the transcription factor known to be in the regulating these hubs. And for instance, keratinocyte-like cells are produced in mimetic TECs, microphore-like cells are produced by SBIB or SOX8 transcription factor. And then tough cells are present with this transcription factor and ciliated cells and so on. So failure of this expression or the failure of the mimetic TECs would cause a multi-organ autoimmunity. And this would lead to some diseases such as the myasthenia gravis. So this process is very important. Now, looking at the single cell and the cell numbers, right? Thymus is where the T cells are maturing, and it is made of mostly thymocytes or T cells. So you can see a very high proportion number of cells is the double positive, double negative, and also single positive T cells. Whereas when you come down where the thymic epithelial cells, for one, these are already sorted EpCAM thymic epithelial cells, and then only we can get these numbers. And even then, it is quite low to detect. So this just shows that the MTEC are rare, these epithelial cells are rare, and these are used for training the abundant T cells through the negative selection. So what approach we had was we screened for all the known human transcription factors, TFs, and they should be expressed in some of the single cell RNA-seq with epithelial cells. And then we looked in our spatial samples for EpCAM positive spots. So EpCAM positive spots means they have an epithelial cell. So if they have an epithelial cells and also express these transcription factors, more likely they are mimetic TECs. So here we assign the ones in the medulla, which expresses EpCAM and transcription factor to be called as mimetic TFs. The rest of the TFs are associated with epithelial cells, and then, we are more focusing only on this subpopulation. So when you do this, and then at bin 50, which is 25 micron, we do get a percentage of EpCAM positive spots expressing these different transcription factors. As I mentioned, AIRE is one of the well-known ones that are expressed around more than 2% of all the EpCAM positive spots. And the other red highlighted transcription factor, like the one like SBIB, which is known for the microfold mimetic TEFs, are also detected. And the ones in blue are the ones that we further show immunofluorescence to say that these are EpCAM and this transcription factor positive. So across all these samples, we saw these rare transcription factors being expressed within the epithelial cells. So overall, this constitutes to only 0.4% of spots that contain the mimetic transcription factor of all the sequenced tissue of the 8 sequenced sample. So as I mentioned before, the epithelial is not 25 micron, it's more closer to 10 micron. So we wanted a way to look at only those spots that are a bit lesser, but then we lost the co-expression of a transcription factor and EpCAM. Sometimes there's a lot of dropouts. So this is where we applied stereosite profiling, where you can, from the same tissue, you can do both proteomics and transcriptomics. For EpCAM, the transcriptomics, you can see how sparsely the dots are. Whereas in the same proteomics, the CD326 is an EpCAM protein where you can see nice structures. Already you can see these are the medulla regions, and it's quite abundant and more clearly defined. So this enables us to do the main thing, that selecting at a bin 20 or 10 micron level, selecting the EpCAM positive spots based on protein, and then looking for the transcriptome, profile for the transcription factors that they are co-expressing in. So we looked at bin 20 for the sites spatial, and almost all of the transcription factors, the mimetic transcription factors that was detected in a bin 50 can be recapitulated at a bin 20 level in the EpCAM spot. So then with immunofluorescence, we show there is a co-expression or co-abundance of both EpCAM and SBIB. This is one of the known mimetic transcription factors, and MyoG, and as well as SOX2. And we also tested some of the mimetic TFs found in the study and found they do co-express with EpCAM and these transcriptions. So what it shows that is these are epithelial cells that are expressing transcription factors that are lineage defining for other cell types like neurons, pancreas, or so on. And another thing we wanted to see was whether these mimetic TFs are in like a hub or in certain regions within the medulla spatially. And so it is known in literature that the Hassel corpels or the HCs are present within the medulla, and surrounding this more mimetic TFs, the transcription mimetic TFs or mimetic TECs are present. So since we didn't have a matching HNE where you can actually draw them out, we used a transcription profile of the spatial and did a proxy of KRT1 expression to find these HCs or where they are located. So KRT1 are MTC304 highly expressed. And in this particular spot, if you can see, you have the cortex and you have the medulla, and there is a region of very low quality. Right? So we didn't get enough transcription reads from this transcription output from this region, right? So in this region, we do find some of the mimetic TFs, and also surrounding this are some of the KRT1 positive sites. So this leads us to believe that these are indeed capturing the corpus. It's just that transcription, to get a transcription output from these sections are a bit more difficult. So and then we looked at the number of the percentage of mimetic TF spots that are present in cortex, medulla, and across the KRT1 high spots, and found indeed they are much higher in the KRT1 high region, which is also associated with MTC4. So then, for spatial co-localization of mimetic TFs, want to see which mimetic TFs are close in spatially close to which mimetic TFs, and then we found that they do form a network based on how much they are co-localized. So if you have two mimetic cells are present quite near each other, and especially the AIRE, FEZF2, and so forming major hubs where with connecting to other mimetic TLs. So for instance, MyoG is connected with AIRE and also with FES2. So they are all interconnected like in a spatial location. They are co-occurring in different cell types. So they do present a different set of antigen presentation happens to the T cells for selection. And then we show this in immunostaining to show that these MyoG in green and AIRE in red are present quite near each other in different regions. We wanted to also see if the transcription factors are associated to any particular tissues or organs. And for that, we saw these transcription factors are from a zebrafish knockout phenotype, what would be resultant in, for instance, MyoG knockout in zebrafish affects the muscle. So then we mapped all the mimetic transcription factors that we found to some of the zebrafish knockout phenotype that is known in a known database, and also to human diseases. So you can see MyoG is for muscle, for pancreas, there are a few transcription factors, and then for brain, you have NeuroD, which represents some of the neurons, and eye, and so on. So this shows that there is a huge repertoire of mimetic TCs representing transcription factor to train the T cells. So come to the end. So in summary, what I've shown you today in this publication is that we profiled spatially and also at a single cell RNA-seq level of human, fetal, and pediatric thymus. Further, we integrated the spatial sample clusters into thymic niches from just transcriptomic profile alone as cortex, medulla, and septa. And then we did find signaling pathways that are active during the positive and negative selection of T cells using cell-to-cell analysis. And then we did identify some of the rare mimetic transcription factors that are in Darwinian TECs, which is around 0.4% of the whole of the tissues that we sequenced. And there we found a higher number of mimetic TECs present near the inferred HCs, which is known and as it is shown here. Finally, these mimetic TECs are spatially present in close hubs so that when they present antigen, they can be presented to the same T cells that is going through the selection phase. Okay, finally, I would like to thank Jonathan Lowe's lab and especially Inchen, Junjie, Pradeep, and Pong for all the work in the study. And here is the manuscript that was published. And also we'd like to thank the NRF funding and A-STAR. Thank you so much, and I would be happy to take any questions.